Welcome to Bioremediation and Dr. Mick. Today we need to explain what are the molecular techniques used in bioremediation. Also we need to explain what are the pathway construction, also non-catabolic genes for the catabolic pathway constructions, and rational enzymes redesign, and finally what are the molecular props. All will be explained shortly. Welcome back. Actually, during the process of genetic engineering, strain the development as well, enzyme redesign, also the pathway constructions, we can undertake the molecular manipulation of the biodegradative systems to improve their utility in bioremediation process. So we have most frequent means of the stimulating of that the bioremediation process and also improvement this bioremediation process. This means including the chemical modifications of the environment can include the addition of the chemicals for improvement and modification, which acts as the electron acceptors or maybe supplemental electron donors, or which enhance the bioavailability. So in such a case you can add some chemicals to modify the environment for the bioavailability. Also we have the physical modification of the polluted sites. This may relate to the ex situ or in situ treatment. Both chemical and physical modifications are sufficient to achieve the bioremediation process if native bacterial populations can destroy these pollutants. Also we have the biological modifications, or what we called before, this is called bioaugmentation. Is the addition, as mentioned, is the addition of the microbes to the polluted sites to accelerate the bioremediation or the biological degradation. So the bioaugmentation is also the one way to use the genetically altered microbes. So we can conclude this means all methods, whatever physical, chemical or even biological, this is to improve the bioremediation process. In such a case, the microorganism able to degrade and also able to construct the pathway for the bioremediation. So now let's go to how the microorganism able to construct the pathway. For the biochemical background for the microorganism, there are several reasons the bacterial aromatic catabolism has received the most attention in terms of genetic manipulations relevant to the bioremediation process. So in such a case we have a very common 5 completely independent routes or pathway of the toluene catabolism have been well characterized for the aerobic bacteria. So to date we have the 5 pathways have been described in the bacteria called the Pseudomonas, despite the fact that at least 2 have been shown to be encoded by broad host range degradative plasmids. So if you look at this pathway, you can find the 5 non-aerobic pathways for the toluene catabolism or toluene degradation. You can find this is the toluene on your left side. So divided, or the microorganism or this bacteria, the Pseudomonas, has the different 5 pathways. The first pathway from the toluene to the cis-toluene, from cis-toluene able to degrade it to the methyl catechol. Another pathway from toluene to orthocresol to the same end product, this is the methyl catechol. The third, toluene to the metacresol to the methyl catechol. The fourth, from toluene able to degrade to the paracresol, then to the protocatechol. The fifth pathway, the toluene can be degraded into the benzyl alcohol and the end product to the catechol. So you can find one organism as Pseudomonas has many pathways for the degradation process. So we need to improve the performance of the microorganism. Maybe we can use some molecular techniques. This figure also one of the examples of the aromatics metabolized through the protocatechols. You can find that the benzoate, the vanillate, ampara, toluene. These compounds can be degraded to the protocatechols, so this compound able to further degraded according to the microbial enzymes. So you can find different pathways. Also, this figure shows some examples also of the aromatic metabolized through the catechol. You can find many products like the phenanthrene, anthracene, the phenol, and also naphthalene, benzene. All of these compounds or aromatic compounds can be degraded to the catechol. So this catechol, according to the microorganism and also according to the enzymes produced by the microorganism, can be degraded to different in the products as this figure. So this pathway constructions depends on the biochemical background. These pathway constructions can be also formed using the Operon deregulation. So the largest single category of the groundwater, for example pollutants on Earth, is the volatile organic compounds, which include the chloroaliphatics. The members of this group they found in the groundwater are the TCE, DCE, and also the vinyl chloride as well. So the regulatory complications arise when environmental application of inducing substrates such as the toluene or even phenol is actually expected. Although these problems may be overcome with the genetically altered variants that don't require inducer substrates. So the related environmental and regulatory complications may be no less formidable. For these reasons, the molecular strategies to overcome these limitations have been applied by a number of some labs. Several compounds like the TCE, co-oxidative enzymes, have been cloned and expressed in bacteria as E. coli. Also for the pathway construction, we have the vectors. The ability to clone, to express, to delete or fuse, and also deliver the various genes in organisms used for the bioremediation is now possible using the variety of some vectors. You know, the vector is one of the gene transfer within the different microorganisms. So in such a case, the plasmid systems developed for cloning and expression inside the E. coli are not particularly well suited to the bacteria other than the Enterobacteriaceae. So we have also the cloning of some plasmids has some limitations. So because of inability to replicate or maybe unrecognized promoter sequences, and also maybe using of the uncommon codon because the difference of the GC ratio, so the plasmid able to replicate in many different bacterial hosts are collectively termed broad host range vectors. So not all the plasmid cloning are successful. So another method for the pathway construction, we can using the hybrid pathways and enzymes as well. So new pathways can be logically built from the components of well-described one. But actually this one also not warranted because all enzymes necessary for the biodegradation already exist. So these are the most of the common methods used for the pathway construction in the bioremediation process. So now let's move to the non-catabolic genes for the catabolic pathway constructions. The first gene of this is called bacterial hemoglobin. bacteria able to producing the hemoglobin due to the presence of the gene production of such a compound as the hemoglobin. You know hemoglobin, this is the blood protein. So we have some bacteria has the gene for the production of such a compound called the hemoglobin. So bacteria such as the Vitricella are obligate aerobic bacteria able to utilizing the carbon source under microaerophilic conditions. And the microaerophilic conditions means this bacteria for growth need the less amount of the oxygen. So this bacteria able to produce the globin protein or blood protein encoded by the VGB gene. This is the gene for production of the hemoglobin. So this protein able to combine with the heme prosthetic prop, this is able to aid in the cellular oxygen. In such a case, able to producing the hemoglobin. The second also, this is called PHB. This is the polybeta hydroxybutyrate. This is a polymer common among the environmental bacteria. Means the bacteria able to produce this polymer. So the gene system of the production of PHB or this compound production has been cloned from the alkagenes bacteria. So this bacteria able to producing PHB as a 3 genes anabolic pathway. So these 3 genes as part of the co-metabolic pathway construct in the cell. So able to catabolize the PHB as alternative to the energy limitations of constructed strains in highly competitive environment. So we have genes or what we call it non-metabolic genes for the catabolic pathway construction. So these genes able to also make the microorganism pathway to enhance and improve the bioremediation process. So now let's move to the rational enzyme redesign. Actually, the site-directed mutagenesis and molecular modeling approaches have contributed to the understanding the enzyme structure and the function as well. So mutagenesis means this is the formation of mutation. For example, the modification of the glutathione reductase enzyme GR of the E. coli, which has led to the alterations in the substrate binding. Also, we have some examples of other recombinant enzymes in which they doing the or perform the alterations using site-directed mutagenesis. So this alteration, this is resulted in the altered the substrate binding efficiencies. So also the altered the rate of the catalysis or maybe stability includes the carbonic anhydrase enzyme. So now let's move to the molecular probes. Actually, we are using the molecular probes to identify some microorganisms as the bacteria or maybe virus according to 2 most important qualities required. The sensitivity and the specificity. Should be sensitive and also should be specific as well. The first probe we have, this is the sensitive and the specific. This is called the bioillumination, called the bioluminescence. Bioluminescence means this is the discoveries that some bacteria able to produce the visible light has been turned into a tool. This tool to the probe, whether certain genes are active in vivo and in situ method for detecting the genetically engineered microorganism, GM, in our environment. So the light produced from the cells can be quantified by the luminometry. This is the light meter. So this light, you can measure this light using this meter. So the light produced will be sensitive and also specific for that microorganism. As an experimental tool, it allows the researchers to amplify exact regions of the genome they wish to study. Also, another probe we have, this is PCR, polymerase chain reaction. Very famous probe. This is the extremely sensitive and also specific method. Such amplified fragments can be cloned, used as probes, or directly sequenced. So even if you need to, for example, you need to check the quantity of, for example, the virus, you can use the PCR. If you want to identify the bacteria or fungus on the molecular level, you can use the PCR and so on. So very sensitive and also specific as well. So the detection by the PCR can therefore be seen as having the advantage of high sensitivity and also specificity as well. Also very speed and also ability to assay many samples at the same time. These are some advantages of using the PCR. Another technique also we have for the molecular probe, this is called immunological techniques. Using the epitope. This epitope actually this is designed to guide the antibody or immune system to recognize the foreign substance in our body. So this technique called the immunological techniques. And this epitope, this is actually particular bacterial strain. One of these techniques is called serotypes. Means this is the test depending on the serum of your blood. So serotyping of the medically important strains, for example, the Salmonella, Streptococcus, even E. coli, you can using the serotype tests or immunological techniques to identify such bacteria. So the same specificity can be exploited in environmental samples as long as the antibody can be found with high avidity to unique surface determinant of the genetically engineered microorganism. So this technique, one such as the antigen-antibody combination, is identified. So the detection of the organism can be greatly facilitated using such a technique. So the cells bearing the epitope of the interest can also be detected by the ELISA. Also ELISA, this is one of the very sensitive and specific technique using the molecular probe. So the immunological techniques, one of the tests belongs to this immunological techniques, this is the serotyping. One of the very famous tests we are using, this is the blood group. Your blood group A, my blood group B, others O, other AB. How did you do this one? You need to 1 drop of the blood to 1 drop of the anti-A or anti-B. This is a serological test. So within this action, between the antibody and antigen, you can detect your blood group. So this is a very famous test and belongs to what we call it the immunological techniques. So now I will show you the table. This table showing or summarizing the molecular methods used to identify and recognize some microorganisms using the molecular probes. So if you look at this table, you can find the different methods from the first column. The method 1, for example, the bioluminescence. This is the novelty. This is the around 5-minute sampling, and also this is able to detect the microorganism in soil, leaves, and other samples. So also has the detection limit around 100 cells per ml if your sampling are liquid, and around 1000 cells per ml if your samples in solid form. Also the method PCR, this is combines the amplification and also hybridization for the detection, And this detection limits also for 10 cells per gram sediments. Also the antibodies, this is the monoclonal antibody MAB. This is the also highly specific. Also you can find detect the limit around 100 cells per spot. There are also another method, enzymatically methods, and also hybridization methods. So this table summarizing the different methods, so you can go through and check the different methods you can find. So also we have hybridization techniques. This is the nucleic acid. The hybridization is one of the most widely accepted molecular techniques used to detect the presence of the particular genes or maybe set of genes in the organisms. So we have two limitations for these hybridization techniques. This is the getting number one, this is the getting enough of the target to be probed. The second limitation, able to determining the optimal stringency of the hybridization. So other limitation also, some environmental samples however commonly contains solids to which bacteria adhere, or maybe which they become entrapped, rendering as well their isolation will be difficult. So this is the some limitation for using the hybridization techniques. So this is the end of this video, and I hope you got the benefit, and please don't forget to share, like and subscribe and activate the bell to reach all of my new videos. Thank you, good luck, and bye bye.